Salmonella enterica is gram-negative, rod-shaped bacterium in the Enterobacteriaceae family. It's a facultative anaerobe, which means it can survive in both oxygen-rich and oxygen-poor environments. This flexibility helps it thrive in diverse places like food, water, and inside animal and human intestines. It's a leading cause of foodborne illness globally, responsible for salmonellosis. People usually get infected from consuming contaminated food or water, especially raw or undercooked poultry, eggs, or produce. The CDC estimates about 1.35 million cases each year in the US alone. Because of its high impact on health, um, Salmonella enterica is important for future healthcare workers to diagnose, public health teams to monitor, and food safety specialists to help prevent outbreaks. Salmonella enterica is rod-shaped bacterium, also known as bacillus. Under the microscope, it typically appears alone or in pairs. It is classified as gram-negative because it has a thin peptide peptidoglycan layer in its cell wall along with an outer membrane. This outer membrane contains lipopolysaccharide, or LPS, which acts as an endotoxin. LPS is responsible for triggering strong immune responses, especially inflammation. This bacterium is motile due to its Petricus flagella, which are flagella spread all over the surface of the cell. These help the bacterium move toward nutrients or favorable environments, such as the lining of the intestines. Some strains also have a thin capsule that provides extra protection and increases their chance of surviving inside the host. Another important structure is the fimbriae. These are short, hair-like projections that help the bacterium stick to intestinal cells. Attachment is the first step in causing infection. Salmonella enterica depends on its capsule, fimbriae, and other outer membrane to survive and spread. Salmonella enterica is a facultative anaerobe, meaning it can grow in both presence and absence of oxygen. This gives it flexibility to survive in range of environments, including the human gut and contaminated food. It produces asexually through binary fission. This is a common method among bacteria where the cells divide into two genetically identical daughter cells. The bacterium is classified as a mesophile. Its optimal growth occurs around 35 to 37 degrees Celsius, which matches the temperature of the human body. Salmonella enterica is a chemoheterotroph, which means it uses organic compounds for both energy and carbon. It gets its nutrients from other organisms or decaying matter. A key survival feature is its ability to form biofilms. These are structured communities of bacteria attached to surfaces like kitchen counters or food processing equipment. Inside a biofilm, Salmonella enterica can resist disinfectants and harsh environmental conditions. This helps explain why outbreaks often happen from contaminated surfaces. Metabolically, It ferments glucose and produces hydrogen sulfide. It does not ferment lactose. These traits help differentiate it from other enteric bacteria during lab testing.
Salmonella enterica has a really flexible genome that helps it survive in lots of different environments. Its main chromosome is circular and ranges from about 4.8 to 5.1 megabases in size. This chromosome includes genes that let it carry out both aerobic respiration and anaerobic fermentation, so it can adapt its energy making process based on its surroundings. It also carries plasmids, which are small pieces of circular DNA that can be shared between bacteria. These plasmids often contain antibiotic resistant genes. That's one reason some strains of Salmonella are so hard to treat. Another important thing is horizontal gene transfer. Salmonella enterica can gain new DNA through processes like conjugation, which is when it transfers DNA directly to another bacterium, or through transduction, which is when viruses carry DNA between bacteria. One of the biggest factors in its ability to cause disease is something called pathogenicity islands. These are large sections of DNA that contain genes for virulence factors. Basically, they give the bacterium the tools it needs to invade host cells, avoid the immune system, and cause infection. Lastly, its mutation rate is moderate, not super fast, but still enough to allow it to evolve over time. This helps it adapt to new hosts and environments and can even contribute to antibiotic resistance. All of this makes Salmonella enterica a very adaptable and sometimes dangerous pathogen. Controlling Salmonella enterica really starts with prevention. One of the most important things is safe food handling. This means cooking meats like chicken all the way through and avoiding cross-contamination when preparing food. It's also important to wash your hands after handling raw meat or being around animals, especially for people working in food production or agriculture. In food processing plants, good sanitation is key. A lot of companies use systems like the HACCP, which stands for Hazard Analysis and Critical Control Points. These systems help them find and reduce risk before the food ever reaches consumers. When it comes to treatment, most healthy people don't need antibiotics. The illness usually goes away in a few days with rest and hydration. But for people who are more vulnerable, like infants, the elderly, or anyone with a weak immune system, doctors might prescribe antibiotics like ciprofloxacin. One of the most important parts of treatment for anyone is rehydration, since the diarrhea caused by salmonella can lead to serious fluid loss. On a bigger scale, public health agencies track outbreaks and investigate sources quickly to stop the spread. So, overall, prevention, proper treatment, and surveillance all work together to control the spread of Salmonella enterica. To diagnose Salmonella enterica, the first step is usually a stool sample. A stool culture is considered the gold standard. This involves using selective media to grow and isolate the bacteria from a patient's feces. Once they isolate it, they can do something called serotyping, which identifies a specific strain based on surface antigens called the O and H antigens. This is really helpful for tracking outbreaks. Another method is PCR, which looks for Salmonella DNA directly and in the sample. It's super fast and very specific, so it's often used in hospitals and public health labs. There's also something called an enzyme immunoassay. That test looks for Salmonella antigens in stool and helps confirm if someone has an active infection. The Whittle test is a blood test that checks for antibodies, but it's not as accurate as the other methods, so it's used less often. And if the infection becomes more serious, like if the bacteria gets into the bloodstream, then a blood culture can be done to confirm it and guide treatment. Overall, between stool cultures, PCR, and antigen tests, there are a lot of reliable ways to detect and diagnose Salmonella enterica.
Now that we've covered the different ways we can detect and diagnose salmonella, let's take a closer look at the biological interaction itself. Our body's defense is a two-stage process, beginning with the innate immune system. Specialized immune cells, like macrophages and dendritic cells, immediately recognize structures on Salmonella's surface, particularly its lipopolysaccharides, or LPS. This recognition sounds an alarm, triggering an inflammatory response. This recruits an army of neutrophils to the intestines to fight the infection. Their job is to engulf and destroy the bacteria through a process called phagocytosis. Salmonella is a very resilient pathogen. If the innate response can't clear the infection on its own, our adaptive immune system is activated. This is a more specialized and powerful attack. Dendritic cells, which have engulfed the bacteria, will present pieces of Salmonella, the antigen, to our T cells. This activates T helper cells, which coordinate a much more effective and targeted assault on the infection. At the same time, B cells are activated to produce antibodies, which are proteins that can neutralize the bacteria and tag them for destruction. On our next slide, the primary affected body systems is the intestines. The infection causes gastroenteritis, leading to classic symptoms we associate with food poisoning, such as watery diarrhea, abdominal cramps, fever, and sometimes headache and vomiting. For most healthy individuals, the prognosis is good. The illness is typically self-limiting, meaning your immune system handles it on its own, and you'll recover within about 4 to 7 days without needing special medical treatment. However, for infants, the elderly, and people with compromised immune systems, the risk is much higher. They can suffer from severe dehydration and other long-term complications. It's also important to remember how this starts. As we see, the pathogen is most often transmitted through contaminated foods like meat, eggs, and even vegetables and fruits. In severe cases, the infection isn't contained to the gut. The bacteria can enter the bloodstream and spread to other organs, leading to a much more dangerous systemic infection. On our next slide, the impact of Salmonella extends far beyond the individual patient. It has a major societal and economic impact. This recall that happened a month ago shows that a single outbreak can lead to millions of dollars in losses for the food industry. Beyond that, there are significant costs from lost productivity when people miss work, and of course, the direct hospital costs for those who become seriously ill. So, how do we approach the treatment strategy? Given that most cases are self-limiting, the primary treatment is not antibiotics. Instead, it's supportive care with a strong focus on rehydration to replace fluids through lost through diarrhea and vomiting. Antibiotics are reserved only for those severe infections or for patients who are in a high risk. This careful approach helps to prevent the unnecessary use of antibiotics and reduces the risk of promoting antibiotic resistance. On the next slide, to summarize, Salmonella enterica is a highly adapted bacterium that directly impacts our health by invading the intestinal system, causing a spectrum of disease that range from mild, temporary illness to severe, life-threatening systemic infection. Second, the significant societal and economic costs of Salmonella, from individual illness to massive food recalls, makes it a primary target for public health surveillance and food safety regulations. Understanding this pathogen is important for preventing disease and protecting public health. On the next two slides are our references. Thank you.